Our group chose to research the caloric restriction theory of aging because of its demonstrated success in preclinical models, as well as the number of biological pathways that reducing food intake can work with. Decreasing caloric intake reduces the amount of glucose release in the blood, which ultimately would increase insulin signaling and transcription, shortening telomere length and increasing intrinsic aging rates. While clinical trial data with humans hasn't yet come to fruition, we will likely see more effects over the coming years regarding GLP-1s and GLP agonists. So two parallel studies that we went over in class from the University of Wisconsin Primate Research Center as well as the National Institute of Aging found that a 30% caloric restriction from middle age led to about a 40% average decrease in age-related death rate compared to the control group. It is necessary to take into account the original differences in weight between the control group monkeys that were between the studies and the monkey species origin, as well as food composition differences between the two places.
The next study looked at the effects of caloric restriction on 48 healthy non-obese adults with the BMI between 20 and 30. The results showed that in the intervention groups, fasting insulin levels were significantly reduced, and there was no change, but there was no changes in fasting glucose in either of the groups, which has been seen to be a biomarker of aging and smaller. The next study looked at the effects of caloric restriction on 48 healthy non-obese adults with a BMI between 25 and 30 and assigned them to one of four groups, a calorically restricted group also doing exercise, and then a very low calorie diet group. Results of the study showed that prolonged caloric restriction lowered fasting, insulin levels, and core body temperature, which are key biomarkers associated with longevity, but there was no significant changes in fasting glucose levels for any of the groups, which have been previously shown as biomarkers of aging in shorter-lived species. In the study, it's important to note that the study was relatively short and only looked at 48 individuals, which is a pretty small sample size.
Another study that we looked at was the calorie study, or using a study using the calorie method. Calorie stands for Comprehensive Assessment of Long-Term Effects of Reducing Intake of Energy. And so, in this study that included 220 adults who, without obesity, were separated into two groups, a group with 25% caloric restriction and an ad libitum control group, and were looked at for a span of 2 years. And so using the calorie method, they were assessed for aging using the Dundon-Pace DNA methylation algorithm, as well as the PhenoAge and GrimAge algorithms. And the results of the study showed that only the Dundon DNA methylation algorithm aging showed that caloric restriction slowed the pace of aging. And so the Dundon pace of aging showed about 2 to 3% reduction in the pace of aging, both at the 12-month follow-up and the 24-month follow-up, both with a p-value of less than 0.003. So pretty significant and
In the aging field, there are a lot of age-related theories out there, and the caloric restriction theory actually relates to a different one, which is the oxidative stress theory of aging, which proposes that the accumulation of oxidative damage contributes heavily to the aging process because as cells produce reactive oxygen species during a normal as cells produce reactive oxygen species during a normal metabolism, it causes cellular damage, but caloric restriction slows down metabolism and thus can reduce oxidative stress by decreasing reactive oxygen species production and increasing antioxidant activity.
So after examining decades of research, I think the takeaway is very clear. Caloric restriction isn't just a diet, but it's a biological signal that slows down the aging process. From yeast to monkeys to humans, reducing energy intake shows measurable effects on metabolism, insulin signaling, and even DNA methylation rates. And the Dundon findings especially stand out, revealing a consistent reduction in the pace of aging by around 2 to 3%. But it's not a one-size-fits-all solution because variability across species, dietary composition, and individual health status complicate the whole picture. And human trials are still in the early stages, and longevity outcomes remain uncertain. Yet, caloric restriction aligns powerfully with theories like oxidative stress, lowering ROS production, and reducing damage to key biomolecules.
All right. I will stop the recording and